All right, we're good to go. So good morning, everyone. Welcome. I'm Richard Hammond. I'm the Chief Technical Officer at Sphere Fluidics. I'm going to take about 15 minutes to talk about some really interesting work we're doing in our platform using pico droplets. I'll give you a little bit of an introduction to the tech, because you need to understand that first, and then the actual applications will make a lot more sense to you. So we'll keep it quick. It is going to be a bit of a canter because I've only got 15 minutes. And particularly we're looking at how we're using our technology to actually do engineering of cell lines and genetic modification. So what's Sphere all about? So we're a company based in Cambridge, UK. Probably tell from the accent, definitely from England. We've been around about 12 years. And our mission is all around finding, analysing and isolating cells, but the really valuable cells that people really care about. So we came out of the University of Cambridge. Originally, the technology came out there, been developing for about 12 years now. As I say, I joined as CTO to try and push the technology forward. We've got a pretty big patent portfolio, lots of clever things we do to make droplets work. Okay, so what's the tech then? We have water and oil emulsions, basically. So tiny little droplets, and the point there is tiny. That's why we call them pico droplets. So each one of those droplets is around 50 microns, 100 picolitres, and they're water in oil. So that's the underlying technology. But then, of course, you can start to encapsulate cells in droplets, and at that scale, it is genuinely one cell per droplet. You then put that into a microfluidic context, and you can start to do lots of interesting workflows with these droplets. So you can merge them, split them. In particular, you can run assays in them and look for responses from the cell, and using that, make decisions. Is that a high producer or low producer? Do I want that one? Do I not want that one? And so you can build much more complex workflows that let you truly assess a cell, decide whether it's a useful and interesting one to you, and move it forward to the next part of your process. And so the slide just shows some photographs of the various functionalities that we have. You can see the droplets being generated. Then you can see the sorting junction where we're moving the droplets around. The far right's an interesting one. There's a dispense function. So we actually put your cells back into tighter plates, microwell plates, so you can move on to your next process. And you can see various images of cells proliferating, growing inside the droplets. A lot of this is actually chemistry, a lot of our expertise in chemistry. You can imagine making a drop that remains long-term stable and allows cells to proliferate and thrive is not entirely straightforward. So it's not just the sort of engineering of it, there's a lot of chemistry behind this process. How does that come into practice? We have two products. There's the Cytomine, which is the world's first fully integrated platform. So often you have the old chip in a lab. It looks great, but it's an entire lab around it. This is bringing all that together and turning it into a true product that you can use. Key things about it, obviously ease of use. You don't want to be worrying about these droplets. Robustness, this is intended for long-term operation, daily operation. And the most important thing about the droplets, you can interrogate very large populations, millions of cells, but you are working at the single cell level. So you get that combination of speed, throughput, but very precise measurement, and that's the real value of the technology, ultimately. Okay, just very briefly, just to get a sense of the workflow integration. Cells, tighter plate, this is how it works. So the first thing we're doing, there we go. First step is encapsulation. So cells in media with oil, forming these droplets, get the flow rates right, one cell per droplet. We then incubate them. Typically, we're looking for a fluorescence type assay, so just to let the assay start working. Then we're sorting, so on the basis of that output. And then we're dispensing. So that's the Cytomine. It takes that entire workflow, runs through it, hands off, it'll do its job. That's what's inside it. Anyone who's done any microfluidics or mass manufacture will appreciate just how complex a device that is. It's taken us some time, but it's got all built-in volume manufacture consumable that does that process. The other platform we call Picomine, and this is much more open. Whereas Cytomine has a continuous process that it runs, Picomine is more open and flexible, and we do a lot of work with our customers here where they want to do something that's a bit more unusual, a bit more funky. We've not tried that before. Can we make it work? This is the platform we use for that. We have a series of custom off-the-shelf chips, but we also do a lot of custom work as well. People want to do some really odd and interesting things, and so we just make what we need to help them do that. Okay. Traditionally, a lot of the work we've done has been in things like antibody discovery, cell line development. But what I'd like to talk about this morning is some of the new things we're doing, looking at cell engineering, some little bit of work in cell therapies, and even some work in synthetic biology. So that'll be the content of the rest of this talk. First question, how long do cells last in these droplets? Because if we're going to start doing an engineering type approach, that does take time, and we need to be confident the cells will survive that. So here's a little bit of data just to indicate that droplets are good for cells, basically. So this is where we took various different cell lines, put them into droplets. These are 80 microns, about 300 picolitres, and basically incubated them for days and days and days. And then every so often we'd take a sample, put them through a nuclear counter, how many of the cells have survived. And you can see from that data that we do start to get drop-off after sort of 72 hours. Some of those cells are starting to die off. But it's not bad. This is no real optimization here. This is just a let's see how long they can go. Certainly good enough to carry on at this point to look more at these workflows. But the good news is cells will survive quite happily in these droplets. As I say, that's due to a lot of work we've done around the chemistry, particularly to allow gas exchange. So the cells are very happy in that environment, even for those extended periods. So the first thing we looked at was transduction. Can we make transduction work? And the important thing here is we are talking doing the transduction in the droplet. It's not that we're somehow doing the transduction and then moving into droplet format. We're actually doing the entire process in the droplet. So this is how we did it. So the first thing we did was encapsulate the viral vector and the cells. And then we incubated them for a bit longer this time, 24 hours. Then we sorted them and dispensed them. Now, these are HTC 116, and we were using a baculovirus expression vector from Geneva Biotech. It's a really nice one because it has two functions. It'll give you GFP transiently expressed. So if you've got it's gone in, you'll get GFP. Then it also does a genome integration and it starts to express mCherry. So you're able to look at both parts of the process and say, has it actually worked and gone all the way through and done the genome change that we're looking for? So these were about 500,000 cells per milliliter, and we were working at an MOI of about 0.25 in this experiment. So that's the workflow. So what did it look like then? So when we ran it, first data on the left is looking at the green fluorescence, TFP. And here we were just looking at peak versus average because we were looking for that bright signal where we've had actually gone into the cell. And we've got about 10% of the droplets were showing a much increased signal, indicating that we have actually put that virus has gone in and started transiently express. Look at the second data, about 35% were showing outgrowth. They did actually survive and continued to grow. And then if you look at the third data, both on day 6 and day 13, we've taken them obviously out of the droplets at this point and they're growing out. We got successful editing. There's the mCherry glowing nicely. And about 3% was successfully edited. Now again, there's been no optimization on this really. It was just let's put it together and see if it works. And so we're very pleased that fundamentally you can use this droplet technology and do this editing process. So we tried it on a few other cell lines. We did that with the HTC 116. We tried a few others, and you can see here both the outgrowth and the positive colonies at the end. MCF7 worked better. The HTC 116 was quite low. But again, this is very much proof of concept, and there's more work we're doing to see how far you can start to push this. But really, this was to demonstrate the idea that you could do this editing within the droplet. And because you can build the workflow, of course, that would then let you move on to much more interesting things, maybe downstream of that, for much bigger workflows. So that was transduction. Then we looked at transfection. Now, for this one, we actually used both platforms. We couldn't get it all to work on the cytomine. That's why I was explaining we have both, because we sometimes do this. If we can't make it work on one, we'll use both. So some of it was on a picomine, some of it was on a cytomine. So here it's a similar process. We're encapsulating cells and a ribonuclear protein. This time we had to incubate it a lot longer, 48 hours, sort and dispense in the same way. Workflows there. Details of the assay. So this was using another HTC 116. This is a Fire one from Horizon. Engineered so it has a mutation, it won't express GFP. And if you do a successful edit using the CAS system, it will start to express TFP. So a fairly simple system, but a good indication fundamentally that process has worked. Before we dive into that, though, the other obvious question was, okay, if it's a transfection process, we're using chemical transfection, is that actually compatible with the droplets? And this is a lot of work we have to do. You know, people want to put new reagents in. Does it actually work in a droplet format? And the good news there, as you can see from the data here, is it does actually work pretty well. So there's the control on the left after 2 hours and 24 hours, and various, typical transfection reagents. And that's just empty droplets. You can start to see how many of the droplets just disappear. They fall apart. And even after 24 hours, we were still getting pretty good droplet retention there. So again, that gives us confidence to go forward with this process. We're not going to actually destroy the droplets, sorry. And again, let's look at some data. So again, we're looking at peak versus average fluorescence to look for that GFP event. And in this case, about 4.5% of droplets we classified as positive. And then again, you can see the outgrowth there, colony 1, colony 2, with the GFP expressing really very nicely. And if you look at that across a number of experiments, a number of 96 well plates, we were getting an average outgrowth of about 16% and a pretty tight standard deviation, just over 3% there. So again, we've done the transduction and now we've shown transfection working really quite nicely, all in the droplet format, actually integral to these tiny little droplets that we're flowing through the system. Okay, now let's move on a little bit looking at cell therapies. And this is really interesting work that we're starting to do. So typically the way we've used our droplets is a cell and something the cell is doing off secreting, some sort of secretion. What we're now moving into is functional assays, so putting different cell types together and starting to assess how they interact with each other. And so again, this was some work we did recently trying to prove that concept and showing it could work. So this is the assay we were looking at. So we're basically looking for the granzyme B. So we have a CAR T cell in this case and a target cell. If the two cells are capable of interacting, they will start to express the granzyme B. That cleaves the peptide substrate and removes the quencher away from the floor, and it all starts to light up. So this is an off-the-shelf assay that we used, a Sense-like kit from Anaspec. But again, this is about moving that into the droplet format. So that's the underlying assay. So again, bit of picamine, bit of cytomine to make this work. The big difference here is at the beginning, we're now co-encapsulating. So earlier I was showing you single cell into droplet. We're now doing 2 cells into droplet. And in this case, we slightly varied the ratio. So we used about 300,000 cells per mil of the CAR Ts and a much higher concentration of the target cell because we want one CAR T and at least one target in each droplet. So you can change around with the ratios to get the measurements that you want. So in here we had about 20% of droplets with one CAR T and 75% of droplets with at least one of the targets, so you can be sure you bring them together. So we co-encapsulate, incubate, detect, and dispense. Before we went too far, we just did some checking of the assay. So this is proof of concept. So this is looking at the CAR T with the control. So the CAR T we were using was engineered to recognize prostate-specific membrane antigens. So that's the difference between the control and the target. Put them in droplets, see what happens. And you can see that up to about 4 hours, we're getting a good difference between the control and the main experiment. If you leave it for too long, we start to get all sorts of background effects and things going a bit awry, probably some apoptosis in there. So we were confident if we didn't take too long about it, it would probably be okay, but too far, not so good. And here's the data. So we put that assay together, and here we're looking at green fluorescence just against size, because we're just interested in what's lit up. And you can see a good population there. And data on the right, both bright field and fluorescence, just looking at the droplets. So we had just over 1,000 droplets, 942 showing fluorescence. So that's about a 28-fold enrichment based on true activity between that engineered CAR T and the target cell. So we're very pleased to see that. Fundamentally, it works. To finish, just a teaser, synthetic biology. Droplets are wonderful. Even better when you can integrate funky measurements. We do a lot of fluorescence. We're now starting to integrate mass spectrometry into droplets. And this is really cool because you take a droplet and you split it. Half goes to the mass spec, gets destroyed, but you get a measurement. And you can use that data to select your cells. it brings a whole new measurement modality in rather than just relying on fluorescence. So this is active work we're doing now. If anyone's interested in that, please come and talk to me. We're looking for people to help us with that and maybe do some more proof of concept type work. That's it. Thank you very much indeed. If there are any questions, we won't have time now. Hopefully that was interesting, and hopefully you've seen what a picodroplet is, what our products are, and more importantly, how you can extend that technology to some really interesting things around genetic modification and co-encapsulation and functional assays. Thank you.